Welcome, sir, and over to you, sir.
Thank you. At the outset, ಮೊದಲಿಗೆ ನಾನು ಕರ್ನಾಟಕ ಸ್ಟೇಟ್ KSTA ಡಾಕ್ಟರ್ ಕರುಣಾಸಾಗರ್, ಡಾಕ್ಟರ್ ಉಮೇಶ್ ಮತ್ತು ಡಾಕ್ಟರ್ ರಾಜೇಶ್ ಅವರಿಗೆ ನನ್ನ ಹೃದಯಪೂರ್ವಕ ವಂದನೆಗಳನ್ನು ಹೇಳಬೇಕು ಅಂದ್ಕೊಳ್ತೀನಿ ಫಾರ್ ಅಲೋವಿಂಗ್ ಮೀ ಟು ಟಾಕ್ ಹಿಯರ್. Thank you. So, beginning with drug resistance in fungi, why we know that fungi, they can cause infections ranging from skin infections like ringworm infection, eye infections like keratitis that can cause blindness, infection of the limbs like mycetoma that can lead to loss of limb, as well as very dangerous infections like mucormycosis, which you know was called as the black fungus. Now this was a life-threatening fungal infection that happened during the COVID-associated COVID-19 pandemic. So the fungi can cause a spectrum of illness ranging from mild annoyances to life-threatening illness. Do we have the count of number of cases of fungal infections that occur worldwide? Yes, we do, and these are conservative estimates. And the latest estimate of 2023 found that around 6.5 million fungal infections happen worldwide. These are serious fungal infections, of whom 3.8 million people die because of these fungal infections. And if you look at the global mortality rate, around 6.7% of all global deaths are due to fungal infections. Now this surpasses the impact of tuberculosis, malaria, hepatitis or pneumonia. And for people who are in research, you know that these are the areas that are actively being funded by governmental agencies. Now despite this burden of fungal infections, fungi are neglected. Having said that, we now know that fungi or fungal infections can cause annoyances to deadly disease, and these infections are treated with antifungals. So what are antifungals? Now antifungals are drugs that belong to 4 classes of chemicals which at very low concentration will inhibit or kill the fungus. Now the 4 major classes of antifungals are the azole group of drugs, the polyene group of drugs, the echinocandins and the nucleoside analogs. Now the azole group of drugs, they inhibit an enzyme called as lanosterol demethylase, abbreviated scientifically as erg11, resulting in less or no formation of ergosterol and subsequent lack of fungal cell membrane and death. Right. Similarly, polyene group of drugs exemplified by amphotericin B lead to formation of cell membrane pores, leading to extrusion of cellular contents and death. The other group, that is echinocandins, they inhibit an enzyme called as beta-D-glucan synthase, which is resulting in formation of the beta-D-glucan that forms the fungal cell wall, resulting in fungal cell death. Another group, that is the final group of major clinically used antifungal nucleoside analogs, they act on the nucleic acid and result in impaired formation of nucleic acid that results in fungal cell death. So why should we be worried about antifungal resistance? Now to tell you that this is a list of all classes of antibacterials, and if you see there is one class alone, cephalosporin. The total number of drugs in this class alone exceeds the total number of antifungals that is available for us for treating patients with fungal infections. So what does this translate to? This translates to the fact that we have very limited number of antifungal drugs, which then translates to it is easier for a fungus to become pan-drug resistant. That is, because there are less number of classes, it can jump from one class to another and with a very short span become pan-drug resistant. Okay. So other issue with antifungals is that antifungals are very toxic, and because they are highly toxic, they can the patient might not die because of the infection, but might die because of the drug that you're giving. Now another problem is majority of the research focuses on Candida, Aspergillus and Cryptococcus. But then other than these bugs, there are several other pathogens like mucorales, Madurella, so on and so forth, which are not covered by many of the global guidelines for their treatment. People are not working on them. So what is antifungal resistance? To answer this question, we first need to look at the ways in which antifungal susceptibility is quantified. And antifungal susceptibility, quantification of antifungal activity is done by methodologies given by two major scientific agencies globally, that is UCAST and CLSI. And in these guidelines, we are able to measure the activity by using two types of tests, one that looks at the minimum inhibitory concentration and the other one by way of broth dilution or agar dilution, and the other one by means of disc diffusion. Now, what the broth dilution methodology gives is something called as minimum inhibitory concentration, that is the lowest concentration of the drug that will inhibit the fungus. Now, in clinical microbiology, these two organizations give us breakpoints. Breakpoints are if an organism's MIC is less than the given breakpoint, it means that the bug is sensitive to the antifungal, and if you use it for treatment, the patient will respond to the antifungal. However, if the MIC is greater than the breakpoint, then the bug is considered to be resistant, and if you give this drug to a patient suffering with the disease, there will be no response, right? However, this is in clinical microbiology. But in research, what is antifungal resistance is if the MIC of the test isolate is greater than the MIC of a reference isolate, then it is called as resistance in research. However, it is not as easy as I am portraying it here. Why? Because antifungal failure in patient is dependent not only on the MIC of the drug but on the patient's condition, and that results in something called as clinical resistance. And clinical resistance is because of clinical factors. If the patient is in shock, and what is the pharmacokinetics of the drug, whether the patient is receiving other drugs which will interact with the antifungal, either resulting in its increased levels that lead to toxicity or low levels that lead to failure, right? And then there are fungal factors, additional fungal factors like resistance, heteroresistance and tolerance. Now just let us have a look at what are these new terminologies or terminologies associated with resistance. Right. So the first one is tolerance. What is tolerance? Tolerance in clinical practice is observed by presence of colonies within the zone of inhibition. You can see these small colonies. Now tolerant cells are cells which are having a transient phenomena where there is slow growth or survival of a subpopulation of fungus despite presence of antifungal. Now, what is the clinical implication of tolerance? Now, the clinical implication, there are not much studies, but there was one study that looked at patients who received the antifungal and who looked at the population of tolerant cells in these patients, and what they found was those patients who had a larger concentration of tolerant cells in their patient isolates, they continued to have infection, which means tolerance also, it's not resistance, but even the tolerant population of cells contributes to failure of antifungal drugs, right? Then there is another concept called as heteroresistance. Now what is heteroresistance? In tolerance, there is no genotypic change, it is just a transient phenomena. Even in heteroresistance, there is a phenomena where there will be a subpopulation of cells which do not have any genetic markers of resistance but will show MICs 8-fold higher than the majority of the population. So even this has been said to contribute to antifungal failure. So drug resistance in fungi is not as simple as it looks in bacteria, because fungi are eukaryotes compared to bacteria. Right. So, how bad is the situation of drug resistance in fungi in India? So, because there are millions of fungi and several hundreds of them cause disease, we cannot cover all of them. What we'll do is we'll look at the resistance pattern of important fungi, important groups of fungi, that is Candida species and upcoming Candida auris. We'll look at another mold that is causing a lot of infections in humans, that is Trichophyton indotini, and we'll look at a globally important pathogen, that is Aspergillus fumigatus. Now, if we look at the 2023 data of the susceptibility, the two most common yeasts that are found in India, that is Candida tropicalis and Candida albicans, are sensitive to majority of the classes of antifungals. But if you look at the other bugs which are known to have higher resistance, like Candida glabrata, Candida parapsilosis, Candida krusei, we find that in Candida glabrata, up to 40%, 30 to 40% of these bugs are resistant to major classes of antifungals like the echinocandins and the higher class of azoles. Similarly, if you look at Candida auris, the situation is atrocious when it comes to Candida auris, which is a major superbug in clinical practice. You can see that only 2.7% of the Indian isolates of Candida auris are sensitive to the azole group of drugs, which were the drug of choice for Candida long time back. However, now the drug of choice for Candida is the echinocandin group of drugs, that is anidulafungin, caspofungin, micafungin, and even in these categories you can see that except for micafungin, the susceptibility is again from 20 to 30%. So similarly is the case for Candida krusei, where high amounts of resistance is seen for echinocandins, that is caspofungin, and a very high resistance is seen for azole group of drugs. If we look at the trends, we see that the two common bugs, that is Candida albicans and Candida tropicalis, their trend is stable and their senses 90%, around 90% of the isolates are susceptible to all the classes of the drugs. And as I emphasized in my previous slides, this is not the same for the Candida parapsilosis, Candida glabrata and Candida krusei. While for Candida auris, as I said, the situation is becoming bad or worse by the day. So, looking at Trichophyton indotini, in dermatophytosis, the drug of choice was terbinafine, and in a study done in 2022, it was found that almost 17 to 75% of all Trichophyton indotini have resistance to terbinafine, while less than 25% of them are resistant to itraconazole, and currently the drug of choice is high dose of itraconazole and longer duration of itraconazole for treating patients with recurrent, relapsing or recalcitrant dermatophyte or ringworm infection. What is the status of antifungal resistance in Aspergillus fumigatus? There are two major aspergilli that cause human diseases in India, Aspergillus flavus and Aspergillus fumigatus. You see the number of cases of Aspergillus flavus in India are higher because in India most of the cases of invasive aspergillosis are caused by Aspergillus flavus. You see that almost 31% of our isolates are resistant to amphotericin B, which was the drug of choice in past for treating all sorts of invasive fungal infection. The same is even worse for Aspergillus fumigatus, where only 48% of our isolates are sensitive to amphotericin B. But fortunately, if you look down, the drug of choice for treatment for invasive aspergillosis is voriconazole, and both our predominant isolates are sensitive beyond 90% for voriconazole. So, what is antifungal resistance due to? Now, antifungal resistance can be of two types: intrinsic resistance and acquired resistance. Now, intrinsic resistance is resistance that is present without any mutations. It is present because it is already present without the presence of the antifungal. It is there, and both acquired resistance and primary resistance are mediated by the same mechanisms like drug transporters, target incompatibility or target overexpression. resistance is acquired in the presence of antifungals, while primary resistance does not need the presence of antifungals and is not meaningful when we are treating patients with fungal infections, because if we know that a fungus is intrinsically resistant to this antifungal, we never use that drug. So that is not the case in terms of acquired resistance, where the fungus may not be known to be intrinsically resistant to the drug. So how do fungi develop resistance? Now fungi, you see here we have fungi that is this is an example of Aspergillus fumigatus, which can reproduce asexually, it can reproduce sexually, and it can also reproduce parasexually. Now if you look at this point, you see azoles. Now if at all there is azoles, there are some amount of cells that are generated because of diversity. In the presence of azoles, these cells that are resistant to the azole group of drugs are selected and they get propagated further. So the azole resistance in Aspergillus is because of presence of environmental fungicides, while in yeast this resistance develops because of overuse of antifungals in the hospital. So antifungal resistance in Aspergillus is not only seen in the hospital, it can also be seen in the environment when the agricultural people use antifungals for crop protection. While the same is not true for yeast, which are predominantly hospital acquired infection, and azole resistance occurs in those organisms because of overuse of antifungals and selection of resistant clones. So how does antifungal tolerance happen? Now one of the mechanisms that has been explained to explain antifungal tolerance is the concept of genome plasticity. Now what happens in genome plasticity is, in the presence of the drug, the fungus, it duplicates its chromosome. For example, in Candida albicans, it is seen that in the presence of azoles, the chromosome number 5 is duplicated. There is one extra limb of chromosome number 5, which results in increased number of ERG11 genes as well as increased number of promoters of ERG11 genes, which results in increased concentration of the ergosterol enzyme, which in turn results in increased ergosterol in the membrane, resulting in tolerance to the azole group of drugs. Similarly, fungi in the presence of stress activate chaperones, heat shock proteins, which will refold the badly folded proteins and make the fungus resistant to the antifungal group of drugs. Now, antifungal resistance mechanisms are mediated for the 4 classes of drugs are mediated mainly by the commonest group is the azole group of drugs, and as I said in the beginning, the azole group of drugs act by inhibiting the ERG11 gene. Now, the ERG11 gene can have mutations, and mutations in these regions, that is hotspot regions, result in reduced affinity of the drug to the ERG11 enzyme, and this results in resistance to the gene. While there can also be gain of function mutation in upstream promoters of the ERG11 gene, that is UPC2, gain of function mutation that results in increased expression of the ERG11 enzyme itself, which can result in bypassing the inhibition afforded by the azole group of drugs. So this I spoke about Candida albicans. Similar mechanisms, that is mutations in the lanosterol demethylase or ERG11 gene, can also result in resistance in other Candida like Candida glabrata, Candida krusei, Candida tropicalis, and the next mechanism of resistance is the drug efflux pumps. Now, drug efflux pumps are mainly of 2 types, and they the drug efflux pumps, the genome plasticity, that is chromosome 3 trisomy, can result in increased number of transcripts of the drug transporter, or it can also result in increased expression of the upstream promoters, resulting in increased expression of the efflux genes which throw out the drug from the fungus into the exterior, resulting in antifungal resistance. So as I said, the resistance is because of either mutations or because of mutations in the drug efflux pump genes itself or in the promoter sequences. So that was about drug efflux pumps. The other way in which the other class to which the fungus can become resistant is the echinocandin group of drugs. Now echinocandin group of drugs, they act by inhibiting the beta glucan synthase enzyme, whose abbreviation or short form is FKS1 and FKS2. Now this mutations in FKS1 and FKS2 genes in Candida albicans, in these regions, if there are mutations, then the echinocandins cannot bind to the FKS1, FKS2 gene, and then there will be, and because they can't bind to it, beta D glucan will continue to be synthesized, and that results in resistance to the echinocandin group of drugs. So as I said, the mutations in the hotspot regions or in the congener, the FKS1 is present in Candida albicans, Candida auris, Candida glabrata and other Candida, while FKS1 congener, that is FKS2, is present in Candida glabrata, and mutations in these regions can result in resistance to echinocandin genes. So the next set or the next class of drugs that I said was nucleoside analog, flucytosine. Now mutations in these genes, that is FCA1, FCY2, can result in resistance to flucytosine. So as I said, there can be mutations in the FCA1, FCAY gene, and that results in resistance to the flucytosine. Now as I said, Aspergillus is a little bit different. Compared to Candida, Aspergillus infection is usually acquired from the community, and if people are using, if in agriculture, if there is use of azole group of drugs for crop protection, what happens is the Aspergillus that is present in the environment gains this mutation, that is TR4 L98H mutation, and the patients can get infected by this Aspergillus containing this mutation, and this can result in resistance to the entire azole group of drugs. Now this is not much of a problem in India, but has become a great issue in the Europe, because in Europe there is heavy use of azoles in the agriculture for crop protection purposes. So how does azole resistance happen in Aspergillus? As I said, there can be mutations. The other name for AIR11 in Aspergillus fumigatus is CYP51A and CYP51B. These two genes again code for the lanosterol demethylase enzymes. If there are mutation in this enzyme, these cannot bind properly to the azole group of drugs, and that results in resistance. Other than this, there can be resistance in the promoter genes, and this resistance in the promoter genes results in overexpression of the enzyme lanosterol demethylase, which again prevents the proper binding of the azole group of drugs and resistance to the azole group of antibiotics. Now some isolates do not have mutations in these genes, but can have mutations in the transporters, the efflux pumps, that is ABC transporters or MSF transporters, and these result in efflux of the azole group of drugs outside the fungus, resulting in resistance. So the azole resistant Aspergillus fumigatus has been described in the world in these regions. In India also it has been described, but it is not as high as it has been in the Europe. So in dermatophytes, I talked about terbinafine, which was used for treatment in past. Terbinafine resistance is mediated by the squalene epoxidase enzyme is inhibited by terbinafine, resulting in death of dermatophytes. However, mutations in the genes encoding for squalene epoxidase results in resistance to dermatophytes. While presence of ABC efflux pumps, overexpression of these ABC efflux pumps due to mutations or due to mutations in hotspot or gain of function mutations in promoters can result in resistance in dermatophytes, and these are the several kind of mutations that have been described to date. I will not get into the details of this because this is a short program. Now, after looking at this, we know that fungi can cause deadly disease and that there are limited options for treating fungi and that they are becoming resistant to this class of drugs, which means that the situation is very bad. But do we have light at the end of the tunnel? Yes, we do have light at the end of the tunnel in terms of newer antifungals which either inhibit other targets than the ones that I described previously, or they inhibit the same target in a different manner. For example, the first drug is ivermectin. Now, ivermectin is found to be active against Candida aspergillus, endemic yeast and pneumocystis. It acts by inhibiting the same FKS1, FKS2 gene that codes for beta-D-glucan synthase. However, its spectrum is higher and it is active against the dimorphic fungi as well. Now, the next one is rezafungin. Now, rezafungin has been shown to be active against Candida aspergillus and pneumocystis. Now, this is a very unique drug because rezafungin has a very high half-life, which means you can give it once a week. All other drugs have to be given either once daily, twice daily, but this is a unique drug which has very long serum half-life, plasma half-life, which means that in patients who had to stay in the hospital to take drugs can now be discharged, and this drug is available both for intravenous use as well as for oral use, and you can use it for OPD patients. Similarly, we have a new drug like fosmanogepix. Now, if you look at the list of susceptibility, a lot of bugs are there in this list of its susceptibility, which are having high intrinsic resistance. These bugs are have high intrinsic resistance, but now we have fosmanogepix, which will act by inhibiting an enzyme called as GWT1, which is present in the endoplasmic reticulum and inhibit the manoproteins. So we now have drugs other than amphotericin B and azoles for treating these fungi that cause serious infections and have very high rate of intrinsic resistance. So the next next drug that is available is opelconazole. Opelconazole acts by inhibiting again CYP51A, but this is also can be given in an inhaled manner and can result in less toxicity. Other drug that is available is olorofil. It also has a same spectrum of activity as fosmanogepix, except that it is not active against Candida and it inhibits dihydroorotate hydroxyl enzyme. Now, another drug that is available is ocetonazole. It is also approved right now for treatment of recurrent vulvovaginal candidiasis. So then another group of drug that is available, in past we knew that amphotericin B was used for was a drug of choice for invasive fungal infection. Now the problem with amphotericin B was that it had to be given in an IV dosing, and when it was given in IV dosing, it would result in renal toxicity and bone marrow suppression. It could not be given in an oral formulation. Now there are newer type of cochleates. What are cochleates? Cochleates are manifolds which fold on themselves, scaffolds which fold on themselves in the presence of high calcium. So what the company has done intelligently is they have sandwiched amphotericin B at the center of this. This is like a BD roll or like a cigarette roll, where in the cigarette roll the tobacco is inside the paper. Here amphotericin B is inside the cochleate. So what happens is, when this cochleate is taken orally, it enters into the bloodstream, and in the bloodstream it enters into the macrophage, which are the cells for activity against fungus. So inside the macrophage, the concentration of calcium is low, and because the concentration of calcium is low, what happens is this scaffold opens up, resulting in release of amphotericin B from inside, and that kills the fungus that has been engulfed by the macrophage. So this is one new type of oral formulation which is now available and could be used for patients with invasive fungal infection. And it has been found to be non-toxic in patients with Candida and Cryptococcus as well as in animal models. So many of these drugs that I told previously are undergoing clinical trials, and many of them are even available in America on now this drug gets into the cell by way of a siderophore and then inhibits the we do not really know what target it inhibits, but once it inhibits the target, it results in inhibition of growth of Aspergillus fumigatus. Now the activity or which is a new drug used for another drug is suprabioleable itraconazole. Now suprabioleable itraconazole, they have changed the shape of the itraconazole, and this has been approved for treating blastomycosis, histoplasmosis and aspergillosis. But in India, people are using this superavailable itraconazole for treatment of recurrent dermatophytosis. So there are studies, though it is still not FDA approved, we are using it for recurrent dermatophytosis. Other alternate approaches for treatment of anti-drug resistant fungi is by vaccination. There are now studies showing vaccination, adaptive T-cell transfer, molecule-based immunotherapy, and use of select antifungal host response compounds like pattern recognition receptors, antibodies, which are still in animal trial stages. They are not yet made to the clinic, but people are frantically working on it. Another set or another area where people are working is the CAR T-cell therapy. And then there are some new targets also, like targets that inhibit hyphal morphogenesis, which get activated in stress response. These are the ones that are now being looked into for antifungal development, and the newer strategies, that is use of artificial intelligence and machine learning for predicting resistance mechanism, optimizing drug candidates. But the problem with all artificial intelligence is garbage in, garbage out. Having said that, there are now studies where people have used these artificial intelligence models for identifying or understanding drug resistance in Candida auris. So another area where a lot of research is happening is by anti-virulence approaches, that is targeting the fungal virulence factors. So if the fungal virulence factors itself are taken care of, how will the fungus cause disease? For example, quorum sensing molecules like farnesol. Use of farnesol is not present in human beings. So if you use a drug that inhibits farnesol, there is no quorum sensing by fungus, and the fungus will not continue to grow, so on and so forth. So other way of reducing antifungal resistance is reducing the use of antifungals in patients who do not need it, and that can be done by use of rapid diagnostics. Now these rapid diagnostic methods, if used properly, will give results in early manner and reduce the use of antifungals empirically. That is, in patients who are really sick, we don't wait for the results, we start the patients on drugs. So if less exposure to drugs, less possibility of development of resistance. Another way that was described by Fisher et al. Was citizen science. That is involvement of citizens in environmental surveillance, where the citizens will pick up samples from the environment, post it to a centralized testing laboratory, where the laboratory will test for genomic markers of resistance, and that will help in surveillance and transmission tracking. It could happen in countries or continents like Europe, and even the Indian Council of Medical Research should look into it. The other way of trying to prevent resistance is the One Health approach. Now, One Health approach, you people all know that health of the environmental health, the veterinary health, and the medical, the human health, they are all related. So, resistance can develop in the environment, get transmitted to animals through food chain, and then come to humans, or vice versa. And if we are able to take a One Health approach, for example, in the case of aspergillus, we know that if in the environment, in the agriculture, if they are using azole-based antifungals, then those can result in development of resistance, and that can be transmitted to patients, and that can result in nosocomial outbreaks. So, if we are able to stop this, if we are able to convince people to stop using drugs that in agriculture that mimic human drugs, we can probably stop this. So what are our future goals? So to prevent drug resistance, or to understand better, there needs to be funding from academia and polity. At this point of time, there are not a lot of robust treatment protocols. We are all following the European or the American guidelines. There needs to be indigenous guidelines, and then definitely there needs to be more research on antifungal drugs and diagnostics. And in case of fungi, there is urgent need for antifungals, because we have less number of classes of antifungals, and probably a multidisciplinary approach could result in wanting to develop new group of antifungals. So as sir introduced, we run a reference medical mycology laboratory here. If any one of you has any isolates that you need to identify, medical isolates, you can contact us on this. Thank you for your patience.